So next up, we're gonna have Dr. Jack Chen to talk about the bio and pharmaceutical track. He is actually a PhD. He received his PhD from John Hopkins University, and he has been an R&D scientist with more than 15 years of experience, focusing on cancer research and drug discovery, and including targeted and immunotherapy. So he's going to talk about, this is actually a new track that we're offering this year, and I feel very excited about this new opportunity. So Jack, please take away.
Thank you, John. If you can click the next slide. So hello everyone, this is Jack Chen. So you may have a question, why biotech pharma track? So like John said, this is an entirely new track. So imagine that back to 2 years ago when the pandemic started to start. So many families are really suffering from the impact of COVID-19. And all over the world, it's a huge damage. And it is who scientists in the lab who work together in the biotech pharmaceutical industry using the new technology that to apply into the lab and identify the new vaccine or the medicine that can help the patient and the people over the world. So the biotech pharma career is very promising if you have a strong passion about the science and want to apply the biomedical research into real drug discovery. So you may have the question, since this is a virtual internship track, so without getting to the lab, what kind of things you can learn? So we will provide the 4 big elements as you list here. You can see that the first one, R&D process. You will acquire the knowledge of the big picture of the drug discovery process in the biopharma industry. And on the right-hand side, target discovery. You will learn how to identify the new target based on the literature review and also the public bioinformatic database. And the third one, preclinical research. You'll get the insight on how to validate the tool compound and clinical candidate in vitro, cell line, and in vivo, such as the guinea pig or the monkey study, before moving to the clinical trial. And at the end, you will receive the training on the leadership in the cross-functional team, which I'll go into the detail in the next couple of slides. Next, please. So what about the qualifications? So if you are a college student or graduate student with a biology-related degree, you are very encouraged to apply this track. And there's a commitment of about 2 hours per week for about 5 months. And if you have a hands-on experience in the lab, that will be a plus, but not required. And of course, you will have a strong passion on biomedical research, and hopefully you can have a creative, detail-oriented, and organized mindset. Next, please. So in terms of the schedule, we will open for the application after the webinar, and then we will collect the candidate application and select the candidate at the end of December. And once the candidate got selected, received the offer, starting January, the step 1, I will introduce you the whole big picture of the drug discovery R&D process 101 for 2 months. And then step 2, try to guide you how to identify new target and how to design the study based on the preclinical research and what is the requirement for the entire drug discovery. And then the step 3 is about in May and June, you will learn how complicated in a matrix environment to work as a team to develop the drug into the clinical trial to help patients with the potential to have a better survival. And in July, you have a chance to showcase what you have learned and present in the team. If you can click next. So here, I would like to show you a big picture what you can learn from the whole drug discovery process. If you can start with the left-hand side, the step one, you will learn how to identify the new target and try to understand how this relevant to the clinic. And then based on the bioinformatic database, trying to identify the target and understand how can we differentiate from other company in terms of new technology. And then step two, you will validate those compound in the vitro cell line or the in vivo mouse model. And then step four, you will learn how to screen hundreds of compounds and narrow down to the lead molecule before the step five, how to select the clinical candidate compound. And then step six, you will work with a cross-functional team as I show on the right-hand side, including the protein engineering group or chemist, the safety team, the clinical pharmacology, regulatory team, commercial, manufacturing, clinical, translational oncology, as well as a legal team. How do we come up together to generate the preclinical document to submit to FDA? We call IND, Investigational New Drug. Before moving to the step 7 and step 8, the phase 1, phase 2, and phase 3. So phase 1, you will start to learn what is required to pass the phase 2, what is the data point we need to collect from the patient in the trial in terms of the safety, pharmacokinetics, PK, PD, pharmacodynamics, biomarker, and then try to correlate with those biomarkers to the patient response. Why some of the patients have response, why some of the patients have no response, and how can we come up with the combination strategy to overcome those patients who did not have response. Next, please. So my final slide, I'll also introduce in this track is that give you a big picture, what's the difference between the preclinical research versus the clinical development. As you can see from the step 1, the compound, how can we generate the most relevant mouse surrogate in a mouse model that is mimic the human clinical candidate. Step 2, the system. So basically work in the lab, you need to identify what is the relevant animal model that we can test by our compound so that we can give the insight how should we select the patient indication. For example, if this is the cancer therapeutics, so are your medicines going to the lung cancer, breast cancer, or other cancer? So this is something you will learn from this track. Step 3, the study design. So in the lab, we all know about roughly the experimental protocol, but how does that difference from the clinical protocol used in the patient? Step 4, people. What are the team members in the research versus the clinical development? Step 5, operation. So we need to have the vivarium to conduct the mouse study, monkey study, but what about in the clinical study? We need to know who we should contact and what kind of instrument, what kind of a protocol in the hospital to make sure we conduct the appropriate clinical trial in the hospital to save patient life. And the final step, you'll learn how is the crisis management in terms of what result is not expected in the preclinical research versus clinical trial, and how can you manage to overcome those and come up with the mitigation plan. So I will stop here and feel free to ask me any related question at the end of this webinar. I'll get back to the job.
Thank you, Dr. Chen. Actually, really thank you not just for your presentation and also your dedication and focus in cancer drug research. Yeah, and actually, if you're not a scientist like me, I'm pretty sure that you have learned a lot from Dr. Chen's presentation on how the whole biotech and pharmaceutical process work.